So our first group of invertebrates is phylum Porifera. This is the most simple phylum of animals, at least that we've classified so far, and this is sponges. So sponges are incredibly simple because they are asymmetric. They don't have any sort of body symmetry. They're sessile, meaning they don't move when they're adults, and they're filter feeders, and they're very, very simple creatures. They have no nervous system, no brain. Um, they don't even have organs at all. They really are just a kind of a cluster of specialized tissues, which is what we're going to see. But they're not really true tissues. Their tissues that they have are a little bit different. They're specialized cells. So they're going to be not quite the same as the tissues that make up your skin or your bone or your organs. There's about 9,000 known species of sponges, and we're always discovering more the more that we learn about the deep sea. These guys are filter feeders, so basically sponges, just like a sponge you use in the shower, you might actually use what once was a real living sponge. You squeeze it and water goes in, it absorbs all that water. You squeeze it again, water squirts out. And so these guys absorb water. They have tiny little pores all over their body where water comes in. And basically, they have specialized cells in there that filter feed out little bits of nutrients in the water. So if we look at the basic structure here of the sponge, you've got most of them have a main opening at the top. That's called the osculum. As you can see in the picture here, these are the osculum, or osculi, I guess, is the plural word for it. And then all of the tiny pores that make up the sponge are called ostia. So those are these tiny little pores that you see, ostia. And the interior of the sponge is made up of these specialized cells and tiny little fragments called spicules. And spicules are kind of like little shards of glass, almost literally, because they're made of silica. And they help to give the sponge rigidity and structure. So basically water moves in through the ostia into the middle part, the middle cavity of the sponge, and that's called the spongiceal, is the term for that, and then exits through the osculum at the top. So the biggest terms I want you to know for this, the ostia and the osculum are kind of the big ones, and then spicules are of course another one. We're going to look more closely at them in just a minute. Sponges can be very complex. Some of them have a lot of canals on the interior of their body, and so I'll show you what that looks like in just a second. But basically, the more different layers of sponge you have, the more surface area there is, and therefore the more food they can absorb. So that's an important part of sponge anatomy. So sponges, again, they lack true tissues. What they're made up of is, I don't want to say false tissues, but basically specialized cells or cells with special duties. So the cells might look kind of like this guy here. So this is a collar cell. A collar cell is going to be attached on the interior part. This makes up the interior cavity of the sponge or the spongiceal. And these guys have a little flagella. And as they move and beat that flagella, they basically help pump water into the sponge. So these guys are like a little water pumps. And then here you see these little tiny fragments are supposed to be food particles. So these collar cells also trap food particles with mucus that they secrete. So that's one type of specialized cell. Amoebocytes, this is another type of specialized cell right here. The amoebocytes will help to do the digesting. So if you remember from biology where you have cells undergo what's called phagocytosis, where they take in little bits of food, they kind of engulf them into a little packet, transport them throughout the cell, and they get used. Well, here, those little packets of food get passed on to the amoebocytes as well, and then transferred to all the other cells in the that make up the sponge. Pinacocytes are another type of cell that are specialized in sponges. They tend to make up the outside of the cell. So these are kind of like equivalent to your skin, or the pinacocytes. Archaeocytes, these guys are special. They actually work kind of like amoebas. They move throughout the sponge. So they would be a cell type that you would see just about anywhere in the sponge's interior. So these guys can help with eating food, transporting food, and then regenerating tissue. Sponges can regenerate. So if you chop off part of a sponge, it'll grow back. So this is an up-close look at spicules. So if you were to look at spicules, if you were to take a sponge and maybe grind up some of it and put it under a microscope, this is what you would see. So the spicules are what give a sponge its rigidity. It's what gives it sort of the firmness And it can also be a protective thing for sponges. So if an animal tries to eat them, the spicules, it's kind of like trying to bite into fiberglass or something sandy. It's really unpleasant. So the spicules are an unpleasant surprise for anything that tries to eat a sponge. There's lots of different sizes and shapes of spicules, kind of like different snowflakes. And so different species might have different shapes of their spicules. So some of them can serve different functions too. Some of them are more defensive versus others are a little more structural. The spicules are mostly made of silica, but they can also be made of calcium carbonate or a flexible protein called spongin. So there's a variety of ways that these things can be made and things that they do. So they're kind of like the skeleton of the sponge. There's basically three types of sponge bodies. So the sponges could be of the asconoid variety. The asconoid sponge is the most simple type of sponge. You have just one interior opening. You have the ostia and then the osculum at the top. It's very simple, very easy. Water goes in through the pores and then out through the top. So they're kind of like a tube with just the one body cavity on the inside. These guys tend to be colonial, so you'll see them in lots of large groups, lots of colonies. The next type of sponge is the little bit more complex. This is called the synconoid. So the synconoid sponge is kind of small here, and you'll see elsewhere on this page on the right side under sponge complexity, you'll see these diagrams more close up. But a synconoid sponge has what we're going to call first degree folding. So you see these little tiny folds on the inside of that body cavity. And that's basically sponge tissue that's folding over and over, and it gives more surface area. So that's going to add basically more area that water can pass over and digest food particles. So it makes it a little more efficient. And then the third type is the leuconoid. So the leuconoid sponge is even more complex. And with the leuconoid, you have even more folding, so even more surface area. So a leuconoid sponge has a very kind of a small or even reduced interior cavity or spongiocele. Instead, it's mostly filled up with lots and lots of canals. So here's a comparison. So on the left here, this is a basic asconoid sponge right here. This would be a synconoid, so you have the first little layers of canals on the inside, and then here's a leuconoid, which is just pretty much nothing but little canals. These are the ones, the leuconoids, that can get really enormous, like this person here is swimming next to one. They can get so big because they have that surface area, so they can transport more water, and therefore they can eat more food. So these are the ones that they're allowed to grow really big just because they have so much surface area and can eat so much. So that kind of summarizes their feeding. I won't go into that too much because we've already described it. They get trapped by the collar cells or the choanocytes, and then the archaeocytes might digest some of that food, and then they get transported throughout the throughout the sponge. As far as reproduction goes, sponges are hermaphrodites. So basically one sponge can have two genders that can produce male and female gametes. They basically just shed them into the water, and then other sponges' gametes cross with theirs, and that's how sexual reproduction can occur. What triggers this is basically changes in water temperature. So it's almost a mechanical trigger that will release the gametes. And so the sponges, again, they don't have any sort of consciousness. They have no nervous system even. And so they're very simple and very sensitive to small changes in the environment. And thankfully, that's what regulates their reproductive cycle, is a change in water temperature. So in addition to sexual reproduction, the sponge can grow, of course, asexually just by cell division, by mitosis, and their cells multiplying, and the sponge itself can grow bigger and bigger. Their process of releasing gametes during sexual reproduction is called budding. So basically, the sponge will develop a little bud that kind of sticks off to the side of the sponge, and it will grow into a new little sponge that will then fall off. Algae actually do this as well, so it's not too uncommon to see this in marine organisms. Okay, so that is all for sponges, and the next phylum we're going to move on to are cnidarians, and that's in the next section.